I'd like to welcome you all to the Lee Jong-wook Memorial Lecture. This is now a series, and I'd like to thank those who endowed it, John and Lynn, who are here with us today. I have a bit of a biography to read. Some of you have been to this lecture series before, and some not. So I'd just like to briefly just let you know who this person is. You can see with the background there that this is someone involved with probably international medicine, and in fact that's true. Lee was born in 1945, obtained his medical degree in South Korea, and he took care of leprosy patients, actually, in his practice. He worked at the WHO for 23 years in varying capacities, and then was eventually named to be the Director General of the WHO. John, who helped me with this, and you'll find out why John wrote this biography, was the first Korean person to be the head of a similarly sized international organization. He died suddenly in 2006 of a subdural hematoma. When he died, the Secretary General of the United Nations, Kofi Annan, mentioned that the world had lost a great man, that he was a strong voice for the right of every man, woman, and child to health, prevention, and care, and he advocated on behalf of the very poorest people. Likewise, President George W. Bush at the time said that Dr. Lee worked tirelessly to improve the health of millions of people, from combating tuberculosis to HIV/AIDS, in his aggressive efforts to eradicate polio. He provided tremendous leadership to the international community as it confronted the challenges of the 21st century, including the threat of an influenza pandemic. His outreach to world leaders and entities increased awareness of potentially devastating public health dangers. I have actually really appreciated these two quotes here. The most important health problem in the world today is the lack of the rule of law. We've been through a pandemic, and I think all of us understand the importance that societal order and law and regulation played in that. And then down here too, people in my country do not live in mud houses for lack of architects. I think that's a witticism, but it actually contains some real truth. This is the connection that I think you might recognize, one of the people in this. This is Lee's family and also Lynn here, and this is in Honolulu, Hawaii. This is where the department's involvement begins. Dr. John Hess met Jong-wook in 1978 when they were both working in Korea, John with the U.S. Army and Jong-wook in an emergency room in a small hospital. Jong-wook and his family eventually lived with Drs. Hess and Stansbury, while earning a master's in public health at the University of Hawaii. That's him, the child. And this is then a lectureship that has been endowed by John and Lynn. I'm very grateful for that, in honor of a very close personal friend. At a time when Jong-wook could not live in either Korea or Japan with his Japanese wife, he lived, basically they endowed Jong-wook's experience at the University of Hawaii School of Public Health, which gave him a scholarship and allowed his family to live there. They lived as a family together, and so this is a real true friend. He referred to the Hess and Stansbury family as his brother and sister, and I think I'd just like to thank John and Lynn, who've been very, very generous to the department in other ways too, for giving us this opportunity. I think what I'll do now is I will move into the honorary lecturer here we have. And I'd like to introduce someone who is known to essentially all of you here, Dr. Josh Lieberman. Dr. Josh Lieberman it fits the purpose of this lectureship. This lectureship is focused on recognizing those who have contributed to the public health through laboratory testing, and I think you're going to see that that's exactly what we're talking about today, or exactly what Josh is going to talk to us about today. Many of you know that Josh started his academic career, or post-secondary school academic career at the University of Maryland, where he got his MD and PhD. Came with us and was an AP and CP resident, and then a co-chief resident too. Then started his journey as an acting instructor, working in Ferric Fong's lab, and then became an assistant professor in 2020, as if there wasn't enough else to do in 2020, and then has been assistant professor in DLMP. He started off in laboratory medicine before that. His administrative roles, he's been an assistant director of molecular diagnosis in the microbiology section, and he's also done some bread and butter lab medicine work, man after my own heart, being the medical director of our Roosevelt Clinic ambulatory laboratory and also point of care testing there too. I will also mention, just in case you don't know Josh is incredibly passionate about what he does. That's evidenced by twice in 2019 to 2020 and 21 to 22, getting the CP Faculty of the Year recognition from our house staff. And also just being incredibly excited about everything he does every time I talk to him about this. And so I'm really looking forward to this talk entitled From Diagnosis to Disease Burden Through Clinical Molecular Tests for Re-Emerging Neglected Diseases. So without further ado, Josh, take it away.
Dr. Baird, thank you very much for that introduction. Stay in the box here. Before I begin, I want to disclose a non-award research collaboration with Anavasi Diagnostics related to work that I have going on related to syphilis point of care development. And I also want to say it's incredibly humbling to be here today. Some of you know, or will by now have guessed, that I overlapped with Dr. Sesson Stansbury at the University of Maryland School of Medicine and was fortunate enough to train with both of them. As a second-year graduate student, Dr. Hess ran into me at a mutual watering hole in Baltimore, it's no longer there, and said, when you're ready to talk about laboratory medicine, come see me. And I think I replied something along the lines of, well, I'm pretty committed to medicine, infectious diseases, or PEDS ID. And well, he was correct, and here I am. Later, I had this really wonderful opportunity to work with Dr. Stansbury and a surgeon mentor, Dr. Sharon Henry, to publish a case series of necrotizing skin and soft tissue infections at the Shock Trauma Center. And that exposure really helped cement my interest in anatomic, not just clinical pathology. Over the years, I've really had a chance to learn a great deal from both of these mentors, including about lab medicine at UW, and hear really some amazing stories, as well as their time with their brother, Dr. Lee, and their collective and important work, in my view, on trauma resuscitation, injury epidemiology, or in the case of Dr. Lee, integrating North Korea into the WHO, and being a fierce advocate for on-the-ground action to support HIV care in Africa, really represents some extraordinary ways to contribute to public health as a physician. And so I guess I'd also add all three strike me as just true Renaissance people. And so I hope that in my small way, I get to contribute and continue that work in my realm of infectious disease diagnosis. I have to confess that while I typically have a great sense that lab med and public health are intimately linked, I actually oftentimes find it very hard to draw those connecting dots explicitly. So I wanted to sort of take a couple of moments to just try and tease those apart. You know, on the one hand, lab medicine is really direct patient care, right? We ensure that patients receive the right test at the right time on the right specimen and provide clinical consults to our patient-facing colleagues. It makes good intuitive sense that good patient care is a key component of improving the health of the public, but I don't really think of this as public health activity per se. Rather, I think those benefits to the public accrue indirectly and after results go out to the patient care teams. At the other end of the spectrum, though, epidemiologic research, in my mind, is often takes place at the national and regional level, thinking about disease trends and patterns, threats and opportunities. This really seems like key elements of public health, and particularly when linked to an outbreak investigation, to vaccination campaigns, sanitation, infrastructure, or publishing evidence-based guidelines based on epidemiologic studies. I think we could also add things like monitoring or discovering pathogens in real time. Most of those activities are actually pretty removed from my day-to-day. So I want to just briefly look at a couple of ways that we can translate those patient results into public health insights. And so I'll start by saying that sort of my early exposure into scholarship in lab medicine really meant learning how to optimize diagnostic pathways, right? Ensuring that what we call laboratory stewardship, really ensuring that the best testing strategy is applied to the right patient. And this is a form of evidence-based medicine. I think it informs, guidelines, but I'm still not sure I'd call this public health. Moving forward, I do think there are ways that we can relate our work, particularly for me as a clinical microbiologist, to local epidemiology. And so one of the examples that I think of is a hospital antibiogram. And so these annual reports, a sort of generic example you see here, summarize the rates of drug resistance in common bacterial and fungal pathogens, which you see listed down the left side, and then drugs across the top, and in the boxes are the percent of strains that are susceptible. This actually strikes me as clearly in the realm of public health, even though I don't think we often term it that way. So, for me, the next question was, how do we really get to national or regional level data? And that's where data aggregation is really the key element. For infectious diseases, this starts with data on specific pathogens and the patients they infect that are accumulated through the process of reporting notifiable conditions. So state and/or federal laws have identified a subset of important health conditions, mostly, although not always, infections, as of sufficient threat to the public that individual laboratories or care providers need to report and sometimes provide samples to local health jurisdictions. So the LHJs then are supposed to generally pass on reports and associated clinical metadata to federal entities like the Centers for Disease Control and Prevention, or may take public health actions themselves. CDC, in turn, can feed back and support those more local or state-level public health activities and produce evidence-based guidelines for how to respond to either emerging threats or proposed treatment guidelines. And the other interaction I want to highlight is with individual labs. So I think we get to contribute to public health activities in the ways we know best, by providing good diagnostic testing. And our lab recently had an opportunity to do just that. Our colleagues at the CDC's Mycotic Diseases branch identified cases of fungal meningitis that were associated with travel to Matamoros, Mexico for elective surgical procedures. Those patients received spinal anesthesia and epidural and were inoculated directly with a potentially deadly mold called Fusarium. We were one of the few reference laboratories that had validated testing available for molecular testing for fungi in CSF, and so we were one of the laboratories to whom patients who were under investigation were referred for testing. And because of that sort of proactive approach, we were able to not only speed results to patients, but also support rapid communication with public health officers. I think that experience with fungal meningitis really highlights a key way in which public need can demand a laboratory medicine response. And so the other way I think, one other example of sort of emergent threats would be Zika virus from 2015. I'm sure we could probably come up with a couple of other examples. And I'd also include neglected diseases in this category. So neglected diseases or neglected tropical diseases are a bit of a term of art. It means sort of forgotten diseases, not necessarily forgotten because they are rare, but generally because they afflict the poorest of the people in the poorest of the countries, so usually low middle income countries. And as we'll see, even outside of low middle income countries here in the US and other high income countries tend to also disproportionately impact at-risk or vulnerable groups. What's interesting about this is that means there's little financial incentive for diagnostic innovation, particularly for companies to move assays, diagnostic assays, through the expensive and time-consuming process of an FDA clearance. So, and also interestingly, labs and clinicians are often not required to report a range of important neglected diseases, which further limits our understanding of these diseases' epidemiology in the population, suppressing demand even further potentially for test development. I think we might call this a market failure, to borrow from our economist friends. In the absence of widely purchasable tests, regulated clinical laboratories like ours can respond to these public health needs by developing and validating their own assays. Such lab-developed tests, or LDTs, play a key role in responding to both emergent and neglected diseases. We in the clinical lab establish the performance of these assays internally, and then every 2 years we open our books to laboratory inspectors to promote scrupulous, high-quality clinical practice. Most of my work really falls in this area, developing molecular lab-developed tests for infectious diseases, particularly neglected diseases. And then because of our laboratory's unique role as a reference lab, we've tried to look at our testing results to learn about the presence and distribution of a couple of these neglected pathogens in the United States. And particularly, I'll highlight two that do not have mandated public health reporting requirements. So I think most people are familiar with this, but just very briefly, I want to remind us that while our laboratories have a unique opportunity to contribute to public health, so you probably know that we are part of a multi-hospital system here in the Pacific Northwest, and we provide care for immunocompromised patients, medically complex patients. We support our Level 1 Trauma and Burn Center and Harborview Medical Center. But we also are the primary reference laboratory for more than 1 million square miles of the US, spanning 5 states: Washington, Wyoming, Alaska, Montana, and Idaho. And we're also a nationally recognized reference laboratory that performs high complexity laboratory testing not offered at other clinical labs in the country. So what this means in practical terms for us is that we see a really fascinating range of cases that come from all over the country. Our laboratory, particularly the molecular microlaboratory, offers a range of lab-developed tests for pathogens, and these fall into really two major categories. On the one hand, we have broad-range PCR assays for bacteria, fungi, and mycobacteria, often with a reflexive next-generation sequencing component that we can use to resolve mixed infections. And second, we have pathogen or sort of groups of pathogen targeted assays. I'll talk about three such targeted assays for neglected diseases today: bartonellosis, leishmaniasis, and syphilis. So these entities share a number of important features that I'll use to highlight the interplay between laboratory medicine and public health. I'd also add that these vignettes reflect my personal journey figuring out how the ways we work, how the work we do in lab medicine can really positively impact the public health. So in June 2021, I was approached by a bright epidemiologic intelligence service officer, Dr. David McCormick, and his supervising medical epidemiologist, Dr. Grace Marks, both at the CDC's Division of Vector-Borne Diseases in Fort Collins, Colorado. They were interested in trying to get a handle on bartonellosis cases in the US. This disease is not a reportable condition, and so there were and remain many key questions about its distribution and potential etiologic agents. So as a little bit of background, Bartonella is a genus of gram-negative bacteria that's difficult to grow in the laboratory. There are at least 7 species that can cause disease in humans, and these are typically transmitted either through body lice or fleas, sometimes other arthropod vectors. Bartonella infections have a number of manifestations. Cat scratch disease is probably the one that's best known. This is a lymphadenitis, a lymph node infection that occurs after traumatic inoculation, often through a cat scratch of Bartonella bacteria. When cats scratch you and introduce this, it's typically Bartonella henselae that's coming from the stool of infected cat fleas. Symptoms include a mild fever and swollen lymph nodes, but other forms can be more serious, such as infections of heart valves, endocarditis, or other vascular lesions like bacillary angiomatosis and hepatic peliosis. So I'll just draw your attention to the pathology here. You see these large dilated blood vessels at the top, bacillary angiomatosis, where there's a fibrotic stroma in the subcutaneous nodule. And then these dilated vessels are sort of embedded in otherwise normal liver trabeculae. And they often form in immunocompromised patients. So Bartonella quintana is the other sort of major species involved, and I need to get to be able to move slides again, and can cause similar conditions, including chronic bacteremia and infective endocarditis. But acute disease can actually be quite dangerous. It can include bone pain, and more severe fever, headaches. This disease is often called trench fever, and fatal infections and severe infections have been reported, particularly in unhoused patients. So we were able to investigate Bartonella epidemiology looking at our 3 tests from our laboratory. The first is a Bartonella bi-species specific assay for the two most common agents of disease, Bartonella henselae and Bartonella quintana. That's a little bit more sensitive for those 2 organisms than our broad-range bacterial PCR, which is also capable of detecting Bartonella as well as other organisms. The Bartonella assay includes a 16S control, so we know if there are likely to be other bacterial pathogens there, including other species of Bartonella. The third assay, in addition to the Bartonella and the broad-range assay that we can use to detect these organisms, is our next-generation sequencing assay that we perform in-house. It's really just when we see mixed infections, and we occasionally tease out things like Bartonella, as well as other important pathogens. We were able to look at over 2,000 submissions for the Bartonella assay, usually with just 1 specimen tested per case. And over nearly 20 years, we detected about 420 cases from all 3 assays. We think this is just the tip of a much bigger iceberg, but does give us a unique look at infecting species, patient demographics, and the anatomic sites that are involved in these infections, I should say. So this graphic illustrates the array of infections these pathogens can cause, ranging from lymph nodes to cardiac sites, joints, visceral organs, and skin. The single most commonly infected organ was the heart, with 140 specimens. We found that more than half, 60%, came from aortic sites, and about 3/4 were from left-sided endocarditis. Only about 8 cases, or 6%, had multiple valve involvement. Most endocarditis cases were detected, interestingly, in males, about 3/4, and were very rarely seen in young patients. In contrast, the second most common site of infection, lymph nodes, tended to come from younger patients, and about 40% from pediatric patients, with about an equal sex distribution. This highlights one really important limitation of the study, and that's the lack of clinical metadata. So we don't know, for example, if the enrichment of males among the endocarditis patients reflects people who are experiencing homelessness, or if there were other common risk factors. So of the 420 cases, we detected Bartonella henselae in about 80% of them, and Bartonella cantana in about 13%. About a third of the B. henselae infections were in lymph nodes, but that accounted for nearly every single one of the lymph node infections, 95%. And then there were about a quarter of the Bartonella henselae infections that were in cardiac sites. In contrast, Bartonella cantana was found almost really overwhelmingly in the cardiac sites, about 83%. This other 7% of cases, these other Bartonella species, are also an interesting mix. So they include several species described only in sporadic case reports, like Bartonella clarigiae, Bartonella vinsonii, both caused endocarditis in our series. There's another very interesting organism with sort of tentative taxonomic status, Canadotus Bartonella meotimonensis. The Canadotus indicates this is sort of still being resolved as to where it fits in the taxonomy. Previously, there's one report of a case report, so we were able to add the second to the list. Bartonella washuensis seems to be more cosmopolitan, but only a few cases have been reported. Myocarditis, endocarditis, and meningitis in the US and Germany. I think we've added the first lymphadenitis case. There is one truly novel species from the lymph node of a child on the East Coast. The remainder probably reflect just the difficulties of identifying organisms. Sometimes 16S, the locus that we amplified, doesn't work for identifying the species depending upon which allele you have. So we had some alleles that we just couldn't confidently resolve as B. henselae or another species. And so we just reported as Bartonella species. I want to add that novel species discovery is actually not confined to Bartonella. This case recently came out and describes a person who had been bitten by a cat and developed this infection on their hand, which ultimately required pretty significant surgical debridement and disseminated to the knee. You can see in this tree, I've sort of shaded this clade right here and highlighted it with a bracket. This is a group of sequences that really are pretty distant from other known mycoplasma species. So this organism was mycoplasma, which is an important pathogen in part because it doesn't have a cell wall, and so it doesn't respond to common antibiotics, beta-lactams, that we might use for these infections. We're now up to 21 cases and counting, and I hope we'll get this work out soon, along with additional work from my colleague, Dr. Trong, looking at sort of mycoplasma distribution in our patients more generally. But I think this is one of those sort of treats that, for me, you don't always get to see sort of new pathogens in your line of practice. And so whenever we do see this, it's a really sort of interesting story, and I think an important piece to get out into the literature. Beyond epidemiology and identifying new species, I think being able to contribute to guidelines is really important. So my colleagues from the CDC, from that Bartonella paper, are now working on culture-negative endocarditis guidelines. They've told me that bartonellosis paper is really informative for that work. We hope to get another case series out on Coxiella burnettii, although actually one of the interesting findings in that is even for this Coxiella, which is a nationally notifiable condition, only a small fraction of those cases actually wind up reported to CDC. We think only about a quarter of them. It's really very gratifying to think that your work can contribute to really more broadly how we take care of patients. We had a similar role for our paper that I worked on with Andrew Bryan on fungal PCR for diagnosis of invasive fungal sinusitis. This project on Bartonella, I think, the most important reason I think I want to share it with you today is that it really opened my eyes to what's possible with our laboratory data. And that's particularly true for a disease that's captivated me for the last 20 years, leishmaniasis. So this picture is from the WHO landing page for leishmania, and it captures, I think, a lot of why I've been so interested in this disease. Here we see a couple of children with scarring lesions on their faces. Kids are often impacted by this infection, the scars really have lasting psychosocial consequences, particularly for girls in countries where there are not a lot of opportunities for female advancement. And I'd point out that these kids aren't at risk for anything they did. They were just born in an endemic area, and often were born in an area where they didn't have a lot of resources, didn't have a lot of economic resources, so they were at higher risk. You know, given that introduction, it may not surprise you to learn that Leishmania has often been called the most neglected of neglected tropical diseases. There are more than 20 species of parasites that infect humans. They're transmitted by dozens of sandfly species. The parasites undergo this complex development cycle in both insect and mammalian hosts, vertebrate hosts, I should say. There are more than 250 million people who live in malaria endemic regions, Leishmania endemic regions. And there are up to about 1 million cases per year, including a few in the United States that are acquired in the United States. The WHO published that this was endemic in the US in 2015. There are generally 3 forms of the disease, involving skin only, cutaneous lesh, involving mucosal surfaces, mucocutaneous leishmaniasis, which can be quite disfiguring, and an organic tropic visceral disease, which is about 80% fatal if left untreated. The infecting species is what really determines which disease you get. So here you see the distribution primarily in the tropics and the neotropics, but selfishly, I want to focus on Brazil. As a much younger student at the University of Maryland, College Park, my mentor, Dave Moser, connected me. He studied Lesh immune responses and macrophage immunology. He connected me with this incredible physician scientist in Natal, Brazil, in the Northeast. So Dr. Selma Geronimo is here second from the end. And she really took me into her lab and under her wing. I spent time in the field collecting peripheral blood samples for GWAS studies to try and understand who cleared visceral leishmaniasis, who cleared infection with Lesh infantum, and who, or I guess L. Chagassi in Brazil, and then who got visceral leishmaniasis, which honestly was an early exposure to laboratory medicine, although I didn't know it at the time. And she gave me opportunities to see a lot of cases of not just cutaneous, but mucocutaneous and visceral leishmaniasis, which, really was a unique opportunity. The 2 women who are standing next to me here, Gloria and Josani, helped make sure that I stayed out of trouble both linguistically and scientifically, and they taught me to run Ficol gradients and count amastigotes and Buffycoat preps that we had collected from patients we had seen in the hospital that day. So I think, needless to say, this really left an indelible impression on me. And then that meant 10 years later, when I was a resident on the dermatopathology service, I saw a patient who had I guess I saw the gross image of the patient's skin lesion, which looked almost identical to this. This image is from the Public Health Image Library. And I was able to convince my attending, Dr. Evan George, that we should get a GIMSA stain, and I took it to the clinical micro lab, and with some input from our senior techs there, was able to identify an amastigote that you see here. I think we saw 2 or 3 in the entire biopsy, which is actually pretty common for New World leishmaniasis, that you don't see a lot of amastigotes. So here's another case in the bottom left that we saw here of an aggressive and persistent nasal septal lesion where there were no parasites seen, versus here is a case of Old World leishmaniasis, L. tropica from Central Asia, that our dermpath service saw a couple of years after that, where there are abundant amastigotes hiding out in these histiocytes. So as a junior faculty member signing out molecular cases, I was frustrated that although our broad-range PCR for fungi could detect these parasites, we couldn't provide the critical species-specific information needed to guide treatment. Here's my oversimplification of that approach, with apologies to the infectious disease docs who are listening. First, we treat visceral disease, just given the risk of fatal outcomes. Second, we treat species that have a high risk for mucocutaneous spread. Those are mostly species within subgenus Viania, particularly Lesh brasiliensis, and we treat immunocompromised patients. Third, cutaneous disease treatment can vary. Small solitary lesions may resolve on their own. Larger lesions or lesions in cosmetically important areas, we do treat. Unfortunately, in the US, treatment is generally toxic drugs that are given systemically. Amphotericin, more recently miltefosine is available. It's actually very hard to access topical therapies in the US, although they do have efficacy in cutaneous lesh. Clinically available testing is still fairly limited. The CDC does the bulk of this work, and my colleagues at Walter Reed test DoD employees and contractors, particularly soldiers who deployed to Iraq and Afghanistan were at high risk for this. Our lab, along with the NIH and the New York State Department of Health lab, have tests online. We happened to go live with our testing quite coincidentally, I assure you, in September 2021, when the CDC closed its doors to clinical testing on a broad agency-wide administrative pause. And they referred testing primarily to us. And that set of circumstances meant that for almost a year, we had this fantastic vista into leishmania epidemiology in this country. So I'll just highlight a couple of high-level results from those findings. I think the first is I did not expect to get as many submissions as we did. Ultimately, in about 186 patients who were tested, half of them were positive. A fifth of them were people under 20, and more than 10% were under 10. We saw a fairly wide distribution of organisms, although clearly biased towards the New World leishmanias, particularly Lesh guianensis complex, Lesh brasiliensis, and a little bit of Lesh mexicana. We saw a couple of interesting species as well, Leishmania lansini, which is a rare species from South America, and Leishmania killicky, which really only circulates in Tunisia. So this does highlight one limitation of our assay. We really can't distinguish, for the most part, between Lesh brasiliensis and Lesh peruviana. Getting within those species complex groups with our assay is quite challenging. It's important to know about that for Lesh brasiliensis complex because peruviana we don't think goes to the, doesn't cause mucosal disease and is relatively confined to Peru, although Lesh brasiliensis also circulates there and has a very high risk of mucocutaneous leishmaniasis. Come back to that in just a moment. I should note that we actually did not get clinical history for very many patients, only about 2/3 of these positive cases. It's actually more than we usually get. We saw mostly cutaneous disease. We saw a surprising amount of mucosal disease, including a relapse. We've seen more visceral cases since then. What I really want to note here, though, is that the cases that we saw and the stories that we saw really highlight the modern landscape, not just of travel medicine, but also the international migration crisis. Right, so we identified cases amongst vacationers traveling to typically Central America, sometimes South America. But we also have detected a number of cases in Afghan refugees fleeing in the wake of American troop withdrawals. And we've seen cases in migrants who have come through or from high-risk areas in Central America. And we know that much of the overland passage, particularly through the Darien jungle in Colombia and Panama, is a really high-risk zone for acquisition of leishmaniasis and for violence. I want to be very careful that I'm not painting migrants as disease-ridden. Whatever one's political opinions are on modern migration, this is clearly a multinational crisis of escalating proportions. And fundamentally, these patients are here. Our providers need to be aware and capable of diagnosing and treating leishmaniasis in this vulnerable population. I should also say there's another really important reason not to just look for leishmaniasis in migrants, refugees, and travelers, right? We have the sandflies and the parasites circulating in this country. Low endemicity is not no endemicity. We did identify Lesh mexicana, which is probably the most common species known and really the key species known to infect humans autochtonously in the US. We didn't get any travel history. So for those couple of cases from Texas that were positive for Lesh mexicana, I don't know where they're from. I'd also add that climate and ecological modeling predicts that the range of both the sandfly vectors and the wood rats that we think are the mammalian reservoirs for these parasites, their habitat will spread in the US, potentially significantly over the coming decades. So this disease is not going away, and fortunately, well, I guess it depends a little bit on the FDA, but fortunately I don't think our test is going away either. I want to say that one of the things we're really interested in is refining our molecular species level identification. So I'll just show you 3 very quick examples where I think we're able to do that within that challenging Lesh brasiliensis complex. Here I'm showing you a part of a tree, just subgenus Viania. Lesh guianensis is down here. I'm not really going to focus on that. I'm going to focus on Lesh brasiliensis complex up here. This first box is clearly identifiable as type 2 Lesh brasiliensis, found mostly in Peru. And we can confidently distinguish this from Lesh peruviana. The second box is also interesting. We've now had several more cases in this clade, and they really seem to all, one, definitely be leishmaniasis, with the attendant risk of mucosal spread, and two, have a geographic association with Central America. We don't have a lot of cases to make that firm yet, but I think it's a very interesting observation that I hope further studies will bear out. I want to also highlight this third box, which is an odd outlier in leishmaniasis. There's only one other sequence like it from Suriname, and I'm really deeply curious. So, the genetic distance here is not huge. You can sort of get a sense of that by the branch length here, compared to sort of other members of this clade, or of this complex. But as my lab works to bring on, my research lab works to bring on direct from specimen genomics to address these samples, I really am interested in exploring this particular isolate to see if it's a novel species, a cryptic species, or some sort of hybrid. Leishmania genomics are exceptionally weird, and given more time, you probably could not stop me from talking about it. I will say that our work doing direct from specimen genomics is very challenging. Most of these cases that we get are from formalin-fixed tissue, which is just brutal for DNA. So what's next for us with this disease? We really want to expand our sample types to include blood. We want to look at other emerging trends like seasonality and co-infections, and think about opportunities to improve our metadata acquisition. Now, I think it would be just a huge disservice to have a talk about laboratory medicine and public health and not address syphilis. I hope that in the following few slides, I'll be able to emphasize the explosive return of this bacterial infection that is transmitted both sexually and vertically and congenitally, highlight a couple of key limitations in our current laboratory diagnostic strategies, and opportunities where lab-developed molecular tests might, and I emphasize the uncertainty here, might help slow this next pandemic. So I think first we have to grapple with the significant rise in primary and secondary cases of syphilis. You can see from this figure published by the CDC in their most recent STD surveillance report, both the overall growth in cases over the last decade, as well, importantly, as some changing demographics with a significant rise among cases in cisgendered women, shown in green, and in men who have sex with women, shown in the darker of the two blues. This is a trend we've seen locally. This graph is from a recent important paper from our own King County, which shows some linear increases in cases among men who have sex with men in blue and tracked on the left y-axis. The smaller scale of the right y-axis for cis women tells us that men who have sex with men have borne the brunt of this disease. However, the epidemic curves for women in King County, shown in red, demonstrate a steep linear, if not exponential, increase in cases over the last several years. It's also worth noting that marginalized racial and ethnic groups not only continue to experience higher case rates than white counterparts, but also increased growth in new syphilis cases. Perhaps unsurprisingly, there is also concurrent growth in congenital syphilis. So these numbers to the left are, I think, already falling out of date, and we're up to about a 10-fold rise in congenital cases over the last decade. And then as you can see on the right, there are systemic causes. Lack of any prenatal care, failure to treat appropriately, but there are also critical failures within the domain of laboratory medicine. So these authors identified two: that lack of timely testing despite timely prenatal care, and late maternal seroconversion accounted for about 20% of vertical transmission events. In another 20% or so of these congenital cases, the authors could not identify a cause for inadequate prevention. Increased cases have landed us in this very vicious cycle. There's increased demand for injectable single-dose and IV penicillin, which has led to shortages for those formulations. Pfizer is telling the FDA, or at least were the last time I checked, that they expect to fix this by mid-2024. But that's still at least 10,000 new infections away. And call me a cynic, but I harbor some skepticism that this will actually resolve by the middle of this year. This is also not new. European and South American countries have repeatedly reported penicillin shortages in the past, and there's global increase in cases and therefore demand. So we are by no means out of the woods, even if Pfizer is able to scale up production significantly. I should also note that the basic biology of T. pallidum is very challenging. It cannot be grown in the clinical lab, for example. The vaccinology and immune evasion strategies are similarly complex and really cool. Those are beyond what I can address today, but I want to note that a lot of seminal work has been done and continues to be done right here at UW. So you may be asking, wait, we have tests for syphilis. What can laboratory medicine offer? And the answer is yes, we have tests, plural, and we have to use multiple tests for diagnosis. So this flowchart, or the couple of flowchart snapshots that I'll show you here, were developed by one of our former residents, Heather Kirkham, I think now a fellow, to help her and her colleagues respond to these consults that she would get asking for help interpreting results. This first image shows just basically how we detect first antibodies against Treponema pallidum, what we call treponemal tests, and then a so-called non-treponemal test, RPR, which serves as a biomarker and potentially a quantitative biomarker of disease. These are reasonably quick assays, but require confirmation. The second chart shows what that pathway looks like at the Department of Health. The quantitative RPR is a highly manual assay and in significant demand, so it is not surprising that confirmation can take 1 to 2 weeks, no matter how talented and dedicated our public health laboratory staff are. You can also imagine that sorting through these multiple results can be a challenge for patient-facing colleagues. Our residents easily take at least 1 call per week asking for help with test result interpretation. Finally, I'll say that point-of-care tests do exist, but they are serological tests that detect treponemal antibodies and therefore don't really help us differentiate active versus past disease. So enter molecular assays. There are a couple of possible use cases. I think some are straightforward, like you take a swab of a lesion and you try and make an organism-specific diagnosis, potentially looking at either protected sites or histopathology and confirming a pattern or a staining pattern or an immunohistochemical stain that suggests there are treponemes present. I think there's some aspirational goals as well, such as screening or potentially resolving ambiguous serologic results. There's also an important public health aspect here. So my colleagues, Dr. Nicole Lieberman and Dr. Alex Greninger, have really been at the forefront of molecular epidemiology of syphilis, and they and other labs have been working on antibiotic resistance surveillance. I want to just try and summarize very quickly a pretty extensive literature on molecular assays for syphilis. I think there are a couple of key themes that emerge for me. The first is there are very, very few assays in clinical practice. There are some common targets. They're all pretty specific. I think we can say that from primary lesions, they're quite sensitive. From secondary lesions, from unroofed skin lesions, the sensitivity is pretty good, but there are a range of estimates. Blood I've put down here with a star because there's some significant pre-analytical variables that impact sensitivity, including how much blood you can start with and what particular fraction of the peripheral blood you go after. So we brought on a lab-developed test here in our high complexity laboratory in September of 2020, and our first year's experience really tells us that this is a niche test. There is a fairly inexpensive assay, or set of assays, our syphilis screen, that works reasonably well. So, it's difficult to estimate our true clinical performance here, but I think it's worth noting that our positivity rate is very low. Looking at the negative results, I don't think that's necessarily because our sensitivity is poor. In about 15% of these cases, we were able to identify an alternative pathogen. We were able to resolve an immunohistochemical stain that was positive for treponemes by identifying a commensal treponeme. Or in a couple of cases, we were able to identify negative serology that was submitted to us along with the patient, or that we had from an in-house patient. From the positive cases, we actually have some very interesting body sites, including identifying a syphilis gastritis, as well as a syphilis aortitis. And so we're looking forward to working with our colleagues, Nicole and Alex, to try and piece together more of the molecular epi and think about potentially what goes where and why. What my colleagues ask for is not a multi-day turnaround test in the high complexity lab. What they really want is a molecular test from blood that we can use for screening that's fast and sensitive. I don't know that we can get there, but we are wrapping up a validation from a large volume blood extraction this month. I think the clinical performance is a big question mark. I will share just a little bit of preliminary data and point out that we're getting 100% detection at sub-100 organisms per mil, down to about 85 organisms per mil in our experiments so far. I think what that translates to in clinical sensitivity is an open question. We have some wonderful colleagues in infectious diseases, particularly Dr. Tara Reid, with whom I hope we can start to address this. I will also say I've been working closely with my friend and sort of scientific partner in many endeavors, Steve Salaponte, on a CDC-funded contract to develop a multiplex point-of-care assay. That's basically ready to hopefully be picked up by a company. But even if it does, it's built off of the clinical assay. Even if it does get picked up, we're still, I think, several years from being implemented. So, we've had a chance to now sort of run through 3 important re-emerging neglected diseases. And I hope I've convinced you that lab-developed tests for these conditions are not only needed for patient care, but that when performed at reference laboratories that can aggregate data, there's a real role in trying to understand the epidemiology of these diseases, and particularly for non-reportable diseases. So bartonellosis and leishmaniasis, I think, really drive this point home for me. I suspect in both cases there's significantly more cases out there than we think. And finally, I want to close, I guess, on a bit of a somber note and just say that really syphilis is the next pandemic. It's here and it's smoldering away. And while more and better lab testing may help, I think this is going to get significantly worse before it gets better. I guess I'll turn away from a more somber note and just want to thank Drs. Hess and Stansbury, who are, amongst other things, great pushers of pastries. God help me if I know when this was taken. I think it was pre-pandemic. I also really want to thank the molecular microbiology and microbiology labs here at the U and at Harborview. Another theme is I need to update my photos. Many of the people in this picture have left, some have come back, and many of the people who made important contributions on the left side over here are not pictured. I also want to recognize my lab. I've managed to land a really outstanding team of people working on a number of research projects, and the many collaborations I've had could not have done this without support from Brad Cookson and his group, from Steve Salaponte, with whom I've, I think, worked on almost every project. Lorenzo Giacconi is a fantastic syphilis biologist who's really carrying forward the legacy of basic syphilis biology at the U here and doing interesting vaccinology work. And made important contributions material contributions, being able to validate our assay. I've had great collaborators at CDC, and on the Leishmania side, Fred Buckner and his research team, Wes Van Voorhees and Selma Geronimo. Worth noting that Wes also has a background in syphilis, working with Sheila Lukart, who really did a lot of seminal and pioneering work in syphilis biology here. And I've had wonderful collaborations with Nicole and Alex and Christina Mara, formerly of UW Neurology, who's done important work on neurosyphilis. So with that, I'll acknowledge my funding sources. I'm particularly thankful to DLMP for opportunities and support for research. And I think with that, take questions and remind you to get your CME. All right.
I'll start with some online questions.
Okay.
So at one point, there were over 80 online attendees. So we have a question from Andrew Bryan attending remotely. So he says, sending a remote applause and, oh, this is more just comment.
Thank you for joining, Andrew.
We'll skip that one. And then I have my own question, actually, which is, thinking about your syphilis work and sort of how to move, what options you have and how to move things, forward. I'm struggling with, hopefully the large volume will get you there, but how do we know what the burden of organism is, or do we have any idea? We do have some idea of what the burden of organism is. There are some, and actually some of the estimates vary, so actually, this was one of the slides I didn't include. I think for primary and secondary disease, particularly secondary disease, we do think there's at least a window where there are reasonable concentrations of circulating either treponemes or treponemal DNA. In one paper that I'm thinking of, I think the median load in blood in secondary syphilis was on the order of about 10 to the 5th. That is several orders of magnitude different from unpublished data that Christina Mara has shared with us, who could only detect in primary and secondary syphilis positivity rates on the order of 30%, and estimated that median organism burden was probably in the low hundreds. So I think this is a real challenge, and I don't know that assay is really going to be impactful. There's some other challenges there as well, in terms of, can we have enough samples on that instrument going through that extraction
that our turnaround time is meaningful. And can we make it inexpensive enough that people are going to use it? So I don't know what that looks like. And I think the low organism burden is a major challenge. It's even worse in neurosyphilis. Dr. Lukart.
I have a question. This is not about anything you've talked about, but I want you to think about something different.
Sure.
So one of the biggest problems in syphilis is being able to differentiate between reinfection and relapse. Any ideas how you might go about looking at that?
Ooh, any ideas about how to differentiate? Any ideas how to differentiate relapse from reinfection in syphilis?
Or just new infection versus past treated infection when the RPR never goes to negative.
Yeah, one of the slides that I moved to the end here was about a serofast state, and so I think that was an easier case because there were no new exposures. But I think when you have someone who is still at risk, doesn't have an RPR resolve, and then as you say, maybe has a new infection now, I think that's extremely challenging. I think that's one case where, again, if you can be analytically sensitive enough with a molecular assay, maybe a molecular assay would be able to help there. I think to really get to thinking about, and so that's for discriminating sort of new infection from just persistent positivity in like a serofast state where your RPR hasn't gone all the way down. I think to discriminate relapse versus new infection, I think is more difficult. And my understanding of the biology when you look at sort of TPRK and these other hypervariable markers, I really don't have a clue how you do that. I think that'd be quite challenging. I don't know, that's something I can chew on, but I certainly don't have an answer on the fly. Yeah, and if you don't have an answer, I'm not sure I'll come up with one. Kate.
You mentioned, or you discussed sort of the challenges around serology and sort of the need for molecular syphilis. I know there are also serologic tests for Bartonella, and what is the role, what do you see as the role and the balance between like serology and molecular for like Bartonella and leishmaniasis?
Yeah, that's a great question. So this was about sort of following up the sort of serology versus molecular question, but for Bartonella, where we do have serologic assays, I would say in Bartonella serology, there are a couple of issues. So one, we often like to compare acute versus convalescent titers, although if you have a rip-roaringly high acute titer, I think you've sort of nailed the diagnosis. There's cross-reactivity across different species. So if you're really interested in trying to understand which organism it is, which probably isn't critical for treating, the molecular is probably an advantage. I think one of the big sort of issues there is, for molecular, we can go after tissue. And so I think if you're looking at weird body sites, molecular may have an advantage. I think Bartonella serology can also can be a little bit challenging to interpret. But again, when you have strongly positive cases, they're pretty strongly positive. I think I really think getting the sense of what organism it is, and particularly if you don't know it's Bartonella going in, my guess is, okay, we got 2,000 cases over a decade and a half or something for the Bartonella specific assay. They're something like, we think, maybe 10 or 12,000 cases per year in outpatients and probably 500 in inpatients. I think people tend to have a good, if people have a good sense that it's Bartonella, they're going to go to serology. I think if you're a little bit less sure, that's when molecular can help. On the other hand, if you can't take a valve out of someone, again, serology is going to be a real advantage.
We have an online question from Susan Butler-Wu, who says, fantastic talk. Do you have any data on your syphilis assay for congenital syphilis diagnosis, which is, of course, a huge issue in terms of increasing prevalence and suboptimal testing?
Yeah, Susan, first, thanks so much for attending. In terms of data on congenital syphilis, no. Our broad-range bacterial PCR has picked up a couple of cases of congenital syphilis, but we've had very few, if any, specimens come through for congenital syphilis with the new targeted assay. I think one of the things that might be a use case for the molecular assay from blood would be to go after cord blood. Trying to think, we had a swab from a I think, a late neonatal presentation, but I think that was in the broad range PCR and not on the targeted assay. I think, I don't know if we're going to see more of this or not. I'm really curious to see how cord blood might perform. I don't know that we know anything about organism burden in cord blood.
And then another online question that's a comment, but I think a valuable comment to discuss if you can, about whether you can study using your molecular assay potentially moving forward to study the persistence of treponemal DNA after treatment, or how that changes over the course of treatment.
Yeah, so could we use our molecular assay to study sort of persistence of treponemal DNA after treatment? I think the question of whether DNA from a pathogen, particularly usually in a cell-free form, hangs around over time is an important question, particularly, and talk about any of this today, but as cell-free diagnostics, cell-free DNA-based diagnostics for pathogens really become more common. There is some data, actually with Bartonella, that Bartonella DNA will persist and be detectable potentially for weeks and may clear slowly. But I think I think it can be a little bit challenging, and particularly we talked earlier about the organism burden in syphilis may be quite low. And so I don't know what turnover is going to look like in that case. Cell-free microbial DNA, I think, is a whole separate issue and one that interests me. And so there may be an opportunity to look at persistence after treatment. And maybe that will help sort of resolve the idea of whether someone was completely treated or not. Dr. Hess?
Dr. Lieberman, how do you bring tests like these to other places, or should you?
Yeah, how do you bring lab-developed tests to other places? Can you or should you? The answer is sort of by their nature, you can't. Because they really, in our regulatory system, they have to be validated by the performing laboratory. So I guess in a sense, if we publish our methods, anyone else can bring this assay on, but they have to have a reasonable complement of specimens to test, either contrived or ideally some true positives. And so someone else could go through a validation. Our methods are published for the leishmaniasis assay, for example, and other people have worked on a variety of lesh assays. I think one of the challenges is for fairly niche testing, right? I mean, we're not getting a lot of samples for these. There's really no reason that many reference labs would want to do this. And there's certainly not a reason that high complexity labs that don't have a broad client base would want to bring them on. I think, in terms of bringing them online at, say, an international site, that's really just sort of conventional science and sharing, publishing primers and sharing data, and then having, offline talks at meetings about how you do things and sharing specimens and things like that. Yeah, great question. Okay. Well, if there are no other questions, I stand between you and treats in E202 next door. Thank you very much for coming.